Assalamualaikum warahmatullah. Assalamualaikum warahmatullahi wabarakatuh. We're going to talk about the molecular pathogenesis of cancer. That this is going to be quite hard topic to be discussed in brief if you're not believing or not trying to put more attentions to it. So I hope that it will lead to answer all the questions about the molecular pathogenesis of cancer that makes you understand more in this topic. So learning outcome. We're going to analyze the the pathomechanism of the cancer from molecular to clinical manifestations. So what is the cancer? Cancer is an abnormal growth of cell caused by multiple changes in gene expression that leading into dysregulated balance of cell proliferations and cell death, and ultimately evolving into a populations of cells that can invade tissues, notice, and metastasize to distant sites, that causing a significant morbidity and, if untreated, once more, if untreated, leads into death of the host. So here are the therapeutic targeting of the hallmark of cancer. You may tend to understand, or you may tend to be familiar with the terms of the hallmark of cancer. What is that actually? So hallmark of cancer is the one that can provides you the quite different characteristic that cancer has, and then it will make you easier to understand on how this cancer works. As you can see, like for example, like sustaining proliferative signaling, that we can use the EGFR inhibitors in order to make those cancer cells cannot have the proliferative signaling tend to existing, keep graduating time by time. So for instance, like for the second hallmark of cancer, like evading growth suppressors, we can tend to use the cyclin-dependent kinase inhibitor. So what I'm trying to portray to you today is that in every types of hallmark in cancer, we have, and we already developed the therapeutic to treat this. So because of the research have been emerging and also growing so much, so that we learning many things about cancer, we studying it more, we tend to look forward for their new characteristics. So in every specific characteristic, all of them has its own function and its own therapeutic targeting. So let's talk about the nature of cancer. Tumor arise from transformed cell by a process known as a multi-step carcinogenesis. So what is multi-step carcinogenesis? In order for a disease to be studied and diagnosed as a cancer, it goes into a several and multiple step. So it's not like a one day here. It's not like happening in one night. It's not an Aladdin that builds a one castles in one night. No. So this carcinogenesis tend to happen that gradually year by year, that probably even in 5 years, 10 years, or even 20 years or more. So let's talk about, for example, in this topic, a normal colon. So in normal colon, we tend to have a mucosa, submucosa, muscularis propria in our body. So what happened is that the germline, such as or somatic mutations of cancer suppressor genes existing, for example, like APC at a 5q21, that these genetic mutations tend to change the function of the normal colon. It changed the structure of the mucosa. It changed the function of submucosa. It changed the constituent anatomy of the muscularis propria, and therefore the mucosa right now at risk. Moreover, that the methylation abnormalities in activations of a normal allele also existing. We're going to call it as a second hit. So what happened in the second hit? The APC beta catenin that goes and also make the proliferation of the cell or the hyperplasia for the cells quite emerging in the normal colon, that in mucosa at risk transform into the adenoma. So in adenoma, they're going to have a proto-oncogene mutations with, for example, like KRAS at the 12p12 that goes it makes into a quite not familiar or even quite differ cells from the previous physiologic cells one, and then it goes into two types. First one is the wild type P53 that will lead into the oncogene induced senescence, and the last one is carcinoma, which leads into the homogeneous loss of additional cancer suppressor genes and the overexpression of COX2 that leads into the additional mutations and gross chromosomal alterations that constituent beta telomerase and many other genes. As you can see, the transformations from the normal colon into the carcinoma is not quite happening in few days. No, quite mistake. So normal colon turns to the carcinoma tends to need quite several years to develop, and then with the several years quite to develop, therefore they tend to make a bigger cells, and then they tend to transport into different parts of the body. That's why we call it as a metastasis. So there's also the multi-step carcinogenesis in the variety of the organ. As you can see that in colon, from the normal into the cancer, it took 5 to 20 years to turn into the adenoma, and then 5 to another 15 years to become a cancer. So for example, like the head and neck, from a normal cells, since with the risk factor of tobacco use, it needs like at least 4 to 10 years to turn the mucosa in our oral and our neck is turned into this dysplastic oral leukoplakia, and it goes into the precancer relation, and then it took nearly 6 to 8 years to become a cancer. For example, like in breast cancer, for instance, like the atypical hyperplasia existing in normal patients, and then it took more than 6 to 10 years to become the DCIS. So in prostate cancer, it took 20 years to become a lesion in precancerous. So what it comes actually, so the etiology of the cancer, we're going to have two different things. First of all, external factors such as environmental chemicals, radiations, and infections. The last one is internal factors such as immune system defect and genetic predispositions. So here are the selected drugs and hormones known to cause cancers in human. I'm going to remind you as well that in the process of a molecular pattern, in order to achieve the carcinogenesis, there are going to be a DNA mutations existing that goes to transform the entire genes in a cell. Therefore, these drugs and hormones who are well known working in sites and changing the DNA of a cells tend to have a probably a similar risk to emerging a cancer as well, or to promote the carcinogenesis. For example, like alkylating agent like chlorambucil or mapillin or nitrosoureas tend to cause a leukemia, or even the, for example, like anthracyclines like doxorubicin tend to have also leukemia. And then for non-steroidal like the ethylstilbestrol tend to have a vaginal or cervix cancer or endometrial cancer and such. So genetic changes in neoplasia, you need to understand, you need to be familiar with those terminologies. What is tumor oncogene? Tumor oncogene is a gene that has a potential to cause cancer, while tumor suppressor gene is a gene whose function is to limit cell proliferations, and loss of whose function leads to cell transformations and tumor growth. For instance, like if you make it easier for you, tumor suppressor gene is like a brake in order to, stopping every little cells to not proliferate that hard or not proliferate under not control. So the proto-oncogene is a normal gene that with slight alterations by mutations or other mechanisms become an oncogene. So before a cell is becoming a carcinogenic cell, it needs to become a proto-oncogene or the normal gene with a slight alteration. So it's not a normal cell, it's just like a cell or normal cell with a slight alterations by mutations that it becomes so easy to turn into the oncogene. So what is inactivations of a tumor suppressor gene? frameshift mutations, deletion of a part of all the genes, and gene slicing by way of promoter methylations. You may notice this, like the mutations tend to have contributions to make a cancer cell. Why so? Because DNA is a blueprint of the function in our body, right? So due to the blueprint is changing, therefore, the cells losing its own function, losing its physiologic function, and tend to be out of control, and then we cannot control the proliferations, we cannot control the divisions, we cannot control the multiplication and such. Next one is the activations of the proto-oncogenes. Mutations, gene amplifications, and overexpressions, or else chromosomal translocations and possibly other mechanisms is quite contributing in order to genetic changes in neoplasia. So here are the examples of the oncogenes and tumor suppressor genes that you may be familiar with, or you may have seen those words in articles or papers or books that you may found, or in lectures that your professor gave you, that the gene tend to have plentiful types. Like oncogene, we can get the genes for growth factors or the receptors like EGFR or ERBB1. It codes the epidermal growth factor receptors and associated with glioblastoma, breast cancer, head and neck, and colon cancer. For HER2, as you may be familiar with, it codes for a growth factor receptor and then mostly associated with the breast, salivary gland, prostate, bladder, and ovarian cancer. So let's just talk another one. Let's talk about the tumor suppressor gene. If there's a mutational in tumor suppressor gene, for example, like in APC, which the function is to step in a signaling pathway, it can disrupt the step in a signaling pathway and leads into the colon and gastric cancer. Or else, like in genes for proteins in the nucleus for RB1, for example, that codes for the PRB protein, a master break for cell cycle. And if the RB1 got mutated, there are no master break for cell cycle. Therefore, which means that the proliferations is uncontrolled. You cannot see that the cells has become quite bigger in size or quite big in amount, and then it leads into the retinoblastoma, osteosarcoma, and bladder, small cell lung, prostate, and also breast cancer. So let's talk about the representative oncogene activated in human tumors. So for oncogene like EGFR or HER1, the cellular function is growth factor receptor that activates the glioblastoma, lung, and breast cancer, that the mechanisms of actions that turn to the oncogene is mutations and amplifications. As you can see also, like for example, like in cyclin D1, that the cell cycle regulator that will go to activating the breast and esophageal cancer, lymphoma, and parathyroid adenoma, that also caused by amplifications and translocations. So if you can see from the table that I'm presenting to you right now, from the right side of the table, so mutations, amplifications, translocations, even if we're gonna talk about the point mutations, deletions, all of these changes in DNA will lead into the changes in the blueprint of how our body works, and therefore we're different and disrupt the homeostasis in our body. So here are the representative tumor suppressor gene inactivated in human tumors. For example, like in P53, it goes for a cell cycle regulator that inactivate the multiple tumor types. And how to inactivate those? It's through mutations. So what happens with them when the mutation exists is lead from many diseases. So yeah, it's quite varying. Like BRCA1 and BRCA2 is a DNA DS break repair, and then it's inactivated for tumor types such as breast and ovarian cancer. It's preventing the cells to turn into those cancer cells, but it got inactivated by mutations and it goes for leading into the familial breast or ovarian cancer. So here are the phenotyping changes in the progressions of neoplasia. We got 8 of them. First of all is genomic instability that goes into the impaired DNA repair and autonomous growth. Second one is enhanced proliferations, goes into the abnormalities of cell cycle control and exaggerate responses to hormonal growth factors, and also aberrant cell cycle checkpoint control. Next one is the evasions of the immune system. It goes for the antigen modulations and masking, elaborations of immune response antagonistic molecules, invasions of tissue and stroma, leading to the loss of cell cohesion, ability to gain access to and form lymphatic and bloodstream will make this cancer cell easy to motility and easy to move and transport to another part of a body. So will goes into the next one is the establishment of the metastatic foci, will goes into the cell adhesion attachment, and then it will goes and make our body to become, having a certain capability to recruit vascularizations to support growth of primary or metastatic tumor. And the last one goes into the drug resistance. So here are the systemic effect of neoplasms. We got vessels compressions that goes into the edema, superior vena cava syndromes, or else like vessel invasions and erosions goes into the bleeding syndromes, or lymphatic invasion that goes into lymphedema. And this is quite varying. And then those quite varying effect can lead into quite different clinical syndrome as well. Let's talk about the epithelial neoplasia. In carcinoma in situ, what goes for this is that they're going to have an invasive carcinoma that goes into the metastatic carcinoma. For example, colon carcinoma, breast cancer, and lung cancer. So here are the schematic depictions of phenotyping transitions of epithelial cells from hyperplasia to invasive carcinoma. I want you to take a moment to highlight the invasive carcinoma, why we pick the term invasive. So I want you to look at this graph in front of you, the left one, which goes into the epithelial cell cancer that is quite, systemic, and then the amount is quite precise. The basement membrane is quite intact, and then when hyperplasia exists or happen, as you can see that the cell from, it's only like 12 cells in the normal parts of a body, but due to the hyperplasia, the amount of cells are increased. So it goes for 16 cells in the normal body. So what happened more and more if it's going to be, keep progressing time by time, that the carcinoma pro-invasive is going to be happening. So later on, it will break the basement membrane, and then it will goes into the invasive carcinoma, and then it can go into the intravascular systems and move into the different places in the body, as well as we call as metastatic focus. So let's talk about the mesenchymal neuroendocrine germ cell neoplasia. It happens in early childhood or young adulthood because these cells are actively dividing and more subject to mutational event. These tumor types may not evolve in specific anatomic sites. Why? Due to the extensive migrations and convolutions of embryogenic cell layers during early development. Sarcoma, carcinoid tumors, testicular germ cell layers, and neoblastoma and teratoma is quite become the examples of this mesenchymal neuroendocrine germ cell neoplasia. For hematologic neoplasia, the malignancies of cells derived from the hematopoietic precursor. The true hematopoietic stem cells has the capacity for self-renewal and the ability to give rise of this precursor, for example, like colony-forming units that proliferate and terminally differentiate towards one of any lineage. Distinct hematologic neoplasms can arise from each of the major cell types. For example, in the bone marrow, it circulates in the bloodstreams and can infiltrate certain organs and tissue. Others may form tumors in lymphoid tissues, particularly lymphomas, which arise from the lymphoblast. So here are the classifications of leukemias according to the cell types and lineage. As you can see, the derivation goes into two. There are myeloid stem cells and lymphoid stem cells, and then the lymphoid stem cells goes into the proliferating mode, what goes into the T lymphoblast and B lymphoblast that, for instance, like if you got mutations on process for turning the mature cells, it goes into the leukemia for this patients. So there are two more staging. The Union International Contre le Cancer, or UICC, and the American Joint Committee for Cancer Staging and End Results Reporting, or AGCCS, are proposing the TNM system. What is the TNM system? I promise you that this TNM system will make you quite easy to identify the patients, whether it goes for the what stadium of cancer for this patient. TNM stands for T is a primary tumor, N is the involvement of a regional lymph node, and the M stands for the presence or absence of metastasis. So it just to the fact that it is quite having a major correlations with the principle of the molecular pathomechanism in cancer, we're going to talk about the principle of the cancer therapy. So the cancer therapy modalities are surgery, radiotherapy, chemotherapy, including hormone therapy, and biologic therapy includes immunotherapy and targeted therapy. So the principles are quite simple. The modern cancer chemotherapy that was proposed by Goodman and Gilman in 1941 is the first administered nitrogen mustard to the patient with lymphoma. So cancer chemotherapy may be indicated as first, primary, second, palliative, third, adjuvant, or even neoadjuvant treatment modality. This treatment with cytotoxic drug is the primary curative modality for a few diseases such as leukemia, lymphoma, choriocarcinoma, and testicular cancer. You have to notice that most solid tumors are not curable with chemotherapy alone, either because of the biology of the tumor or because of advanced diseases at the presentations. Chemotherapy in this setting is often initiated for palliative purposes. It is often possible to decrease tumor size or to retard growth enough to reduce untoward symptoms caused by tumors. So first of all, most of these anti-cancer agents are interfere with the cellular synthesis of DNA, RNA, and proteins. So here are the mechanisms of the actions that tend to be used for make you easy to absorb the figure. I wanted to show you that in every little part of the DNA or RNA productions or even amplifications, there is going to be inhibitions in every parts of the DNA, like we're going to do for the purine synthesis or ribonucleotides, or how to create deoxyribonucleotides, that every little parts that possibly can be attacked by mutations, we can obstacle this process. So anti-cancer agents tend to use in cancer chemotherapy are commonly categorized by their mechanism of actions or by their origin. The alkylating agent exert their effect on DNA and protein synthesis by binding to DNA and preventing the unwinding of the DNA molecule. Why? Because the anti-metabolites resembles naturally occurring nuclear structural components or metabolites such as the nucleotide base or inhibit enzyme involved in the synthesis of DNA and proteins. Anti-tumor antibiotics derive their name from their source. They are fermentations products of Streptomyces species. So here are the examples. There are anti-metabolites, alkaline agent, microtubule targeting drugs, topoisomerase inhibitors, heavy metal compounds, miscellaneous agent, and endocrine therapy. For targeted and biologic agent, there are monoclonal antibody, there are cell surface glycoprotein, monoclonal antibody, agent that targets growth factor receptors and ligand, and biologic and immune therapy. Let's talk about the multiple toxicities related to anti-neoplastic drugs. Drug-specific toxicities such as doxorubicin-induced cardiotoxicity and bleomycin-related pulmonary toxicity may lead into several adverse effects, includes nausea, vomiting, myelosuppressions, mucositis, alopecia, infertility, and the last one is carcinogenesis. Thank you, and I hope you are entertained, and then it will broaden your horizon about the new insight regarding the molecular mechanisms of cancer. Thank you.